Hello, welcome to Cognitum, a show dedicated to the present and future of science and technology. I'm your host, Iosif Gershteyn. Hello, and welcome to Cognitum. Tonight we have with us Victor Goldmacher. He is the co-founder and chief scientific officer of Emuvia, formerly one of the founding scientists of ImmunoGen, and professor at Harvard Medical School. Victor, welcome.
Thank you.
Victor, could you tell us about your academic work during your postdoc at MIT and when you were a professor at Harvard Medical School?
When I started my postdoc, actually, I wanted to study cell biology a little bit more because I wasn't much familiar with that. My background is chemistry and my PhD is in biochemistry. So I came to a lab that studied mutations, mutagenesis, and DNA and how cells repair DNA, because actually in our cells DNA is damaged all the time from radiation, from many different things. Just from mistakes, random mistakes, because actually when DNA is replicated, which means that it's copied when cells divide, errors are made, and then it is repaired. So we studied mechanisms of DNA repair, and actually, accidentally, I found a DNA repair mechanism that previously was not known in any eukaryotic cells, which means any cells of such as yeast or plant cells or mammalian cells or animal cells. It was only known in bacteria, but not in anywhere else. And accidentally we found it in human cells, and that is called mismatch repair. Mismatch repair means that when DNA is copied, sometimes a wrong building block is put in, and then DNA, it's a kind of a dimer to a spiral that actually consists of two chains of nucleotides, and in this place where it's a mismatch, it's a bump. So typically it's smooth, and in this place is bump, and then a special enzyme crawls along this DNA, and when it feels that there's a bump, it replaces that with the right building block. Well, of course I simplify, but that's essentially what it does. And actually we found that, and then it took me a couple of years to publish this paper. And what is amazing is that the science biology advances so fast that experiments that I had to do for almost 3 years now could be repeated within a week.
Wow, what changed that it took 3 years?
We have new tools that we didn't have then, and many things that I had to do myself and to do it in the lab. Now biology is that many tools you just buy ready to go. You buy this, you buy that, and you solve your biological problem. And it's very, very powerful. And this process is accelerating. It's actually exponential. Biology becoming more and more powerful. Mm-hmm. And these days, you don't divide people into cell biology, molecular biology. No, you are a biologist. You use all tools that are available to you to solve your biological problem. And you should be versatile in many different things if you really want to succeed in solving this problem.
Mm-hmm. And what did your research lead to?
Well, actually, it was it's just a building block and a kind of wall, a brick wall, which is science. So right now, it's already lost. It's somewhere down below. But what happened actually? I isolated a cell line that has a defect in this type of DNA repair. That's how we found it. And then after that, many scientists started using the cell line. We shipped it to many people, and they studied that, and they found some interesting things. And actually, Now it's well known that this type of DNA repair exists in human and other mammalian and actually other animal cells. But, you know, they no longer cite me, they no longer read my papers, because actually in biology especially, all moves very fast. Within 10 years, it becomes unrecognizable, and all the papers are no longer very interesting. Well, it's a generalization. Some papers are, but some papers are not. Most papers are not, because people already know about that. It became knowledge, and they move on. Like, for example, even Newton. Nobody really reads works of Newton anymore. You don't need to, because actually it's now well known, and now you read modern textbooks if you really want to study it, because it's no longer science, it's actually already acquired knowledge. But you don't read Newton because it's almost incomprehensible, because then he didn't know what we know now. We stand on shoulders of the giants. That's how they say. That's why we see better and further.
So what did you do after academia?
You mean after Harvard Medical School?
Yes.
So that was actually at MIT. Then when I joined a group at Harvard Medical School, and I wasn't really a professor, full professor, I was assistant professor, junior faculty, and I was part of the group that we were at Dana-Farber Cancer Institute, which is a part of Harvard Medical School, and we were developing anti-cancer drugs. And that was targeted therapy. We were collaborating and part of a larger division, tumor immunology division, that the head of this division was Stuart Schlossman, who was a pioneer of studying monoclonal antibodies in the United States. One of the first people, monoclonal antibodies were just discovered, I don't remember, 1977 or 1978, and by the beginning of 80s, he started studying them and applying to mostly blood tumors. And it was really powerful because he found many very interesting antibodies that advanced our knowledge of blood tumors. And actually, we as a research group could also use these antibodies trying to target certain drugs to these cancer cells in a hope to kill them. So that's what we did, and then it became clear that we have some interesting results, we have some interesting drug candidates, and at this point a company was formed, ImmunoGen. And we went to the other bank of Charles River, to Cambridge, and became a company.
And what did you do at ImmunoGen?
Basically the same thing. We tried to discover and then develop targeted cancer therapies on the basis of antibodies. So basically, antibody is a protein that recognizes a certain other molecule. And there are many molecules on cell surface, and some of them are called tumor-associated antigens, which means that basically these molecules are overexpressed on the surface of cancer cells and not so much, not expressed that much on normal cells. That means great. You can take this antibody and you inject it into a patient, into bloodstream, and then it will preferentially bind to a cancer cell. So if the cell is killed, fantastic, you start treating cancer. The problem was, as we discovered later, is that naked antibodies, just antibodies, yeah, they bind to cancer cells, but only very few of them then help to destroy cancer cells. Most of them simply bind. So you have to do something else. You have to attach something toxic to these antibodies. So that's what we did. I went to the clinical director of Dana-Farber, and he gave me a list of 300 very toxic compounds that NIH screened. So I gave it to our chemist and said, look through these compounds. Anything that you find interesting in terms of chemistry that you can attach to an antibody. And he found one compound that the name of this compound is mitanzin, that could be modified and then attached to an antibody. And that's what we did. And the resulting drug we called antibody-drug conjugate, because we conjugated a drug, cytotoxic compound, to an antibody. So now what we hope would happen, antibody would selectively bind to a cancer cell, then internalize inside the cell, and then this drug will kill the cell. Well, little did we know, actually, it took us many, many years. We published our first paper in 1992. Then second paper in 1996. That was the very first 2 papers in the field. But then other companies followed us, and actually one of them made a successful antibody-drug conjugate that was approved by the FDA, Federal Drug and Food Administration, even earlier than ours. It took us many years before our first antibody-drug conjugate was approved. So the first paper was published in 1992. We started studying that in the 80s, and the first antibody-drug conjugate that we made was approved in 2000, I don't remember, 13, I guess. So a long time. The second one that we developed was approved recently, just last year. Also, accidentally, I discovered an antibody that just by itself destroyed cells. So we did some entirely different experiments. I wanted to prove that a professor at the University of Texas was wrong. She published a paper, and I felt that it was wrong. So I tried to reproduce her studies. And indeed, the studies were not reproduced, but an antibody that I thought was a negative control killed cells very well. I was very impressed. And then it took us several years, and we developed that antibody. Then we licensed that antibody to a company, pharmaceutical company, Sanofi. And now it's one of the best drugs, I think maybe the best or maybe second best, for treating multiple myeloma, which is a very aggressive blood cancer.
So from your work there are now 3 FDA-approved drugs.
That's correct.
Mm-hmm.
And so that's what I did, actually, many, many years, and there are 3 FDA-approved drugs. But that's not me. It's actually, I was part of the team. I played an important role, I think. I think that for example, I accidentally discovered this antibody, and then I then studied its properties with my group, and then another person came to my group, a former, he was at MIT as a graduate student, very talented guy, and he took over, and without him nothing would happen. But he developed this molecule further, and then after that, Sanofi was very interested, and we gave it to Sanofi. But then there was another problem. At that point, a German biotechnology company did something wrong, and in a clinical trial, several patients, I think, died, and some other patients, there was a lot of damage. And then after that, they became FDA became very scared. So the clinical trial of our antibody had to start from very, very low dose. Everybody knew that this dose will not be therapeutic, will not do anything, but FDA wanted to make sure that it will not be toxic to the patient. Who wants to go to a clinical trial when they inject you a molecule which is useless because concentration is too low? Nobody wants to. And that's why this clinical trial took many, many, many years. But gradually doses went up and up and up. Nothing really happened. It's a very safe antibody. And finally, they came to a therapeutic dose. And now it's a very powerful drug against multiple myeloma.
So in terms of the drugs currently available, what are the different modalities of cancer therapy that are on the field as of 2023?
Well, it's still the most important ones. For solid tumors, it's surgery when you, what they call in their jargon, debulk the tumor, because sometimes there are massive tumors or sometimes there are many tumors. For example, in ovarian cancer, there are many, many, many micrometastases that align the abdominal cavity. So a surgeon goes there and one by one pick up most of them. But of course some of them are left behind. Then after that, after surgery, not always surgery, it's chemotherapy, which actually you give the patient a very toxic drug that kills cancer cells and also is toxic for normal cells. But a good chemotherapeutic drug kills cancer cells first at a lower dose when it really becomes very damaging. So basically it's given at what is called it's slightly below maximal tolerated dose, so it's relatively safe for the patient, even though it's also very damaging to his or her body. And then if it's successful, some cancers are cured. Other cancers could become a chronic disease for many, many years, which is also great. If a cancer becomes a chronic disease with almost no symptoms, and you can maintain a patient for 10 or 20 years, sometimes it's great, because actually then maybe this patient is an old patient and he will live all his or her life before dying of cancer. So chemotherapy, also radiation. There are many types of radiation, and X-rays, of course. Also, they implant some radioactive compounds with certain cancers, and that's alpha radiation, which is very local, alpha particles that travel very short distance, so it's relatively safe. So they implant it right next to a tumor, especially prostate tumor, and then it just irradiates prostate tumor, but nothing else, essentially. Or neutron beams, which are very focused, and again, they can, it's used, for example, for prostate cancer, eradicate prostate cancer, focused exactly on the tumor. Then there are newer treatments, but these are the standard treatments: chemotherapy, surgery, of course, and radiation. And it's used both for blood cancers, which are also called liquid cancers because it's in the blood, which is not exactly true. It's not exactly liquid, but these are cells that travel in the blood or stuck somewhere in the bone marrow. Or solid tumors, which are tumors of some other internal organs. And so antibodies are a new type of treatment. Some antibodies are very, very effective, and they are much less toxic. For example, Herceptin is an antibody that is used for breast cancer, and it helped a lot of patients. But not all breast cancers are responding to Herceptin. I think that's like 20-30% of breast cancers. And people know which ones respond, which ones don't respond. And you can analyze that beforehand, pre-screen the patients. Also, there are many, especially useful antibodies against blood cancers and that treat lymphomas. And sometimes these are the antibodies that can keep people relatively healthy for many, many years.
What do you think are the most promising future directions of cancer therapeutics?
Well, now there are, what I didn't mention, there are several directions. And I wish I knew which is the best. Which is the most promising. Nobody knows that. The power of modern science especially is that it's a huge army of people who each of these people work in a different direction, and beforehand you really don't know what will be the most promising one. But because they work in all directions, some of them turn out to be phenomenal. Others are not interesting, don't work as well, or don't work at all. But now we know what works, and now everybody abandons everything else, jumps on this direction, and then quickly advances that. And what is amazing with biology that it makes new discoveries, and biomedical science especially, faster and faster and faster, exponentially. So it all started actually, I think, in the 60s, 70s, modern biology and modern biomedical science, which is related to the discovery of genetic code and structure of DNA, and also new types of chemotherapy. And now I think that there are several directions which might be amazingly good. So one of them is antibody-drug conjugates, as I mentioned. It has certain limitations, but also some of them became very effective and much safer than chemotherapy, because chemotherapy is toxic. Antibody-drug conjugates are also toxic, but much less toxic because they are targeted. They target cancer cells. And that means you can give it to patients at a lower dose, and they will tolerate it better. Previously, antibody-drug conjugates were made with cytotoxic compounds. But now it's a very interesting new type, which is called PROTAC. And that is a conjugate of antibody, and then a certain molecule is attached that goes inside the cell, goes to a protein which is called oncogene, or cancer-related protein, which actually helps cancer to progress. And then it tags it in a certain way, so that the cell actually now targets this protein for destruction. And all of a sudden, very quickly, these proteins are destroyed, and the cancer cell cannot function. But in normal cells, these proteins are not expressed or unimportant because they don't proliferate, and then it becomes even safer. So that's one direction. Another direction is immuno-oncology. Immuno-oncology is an umbrella term, really. There are many different things, but basically you engage certain types of immune cells to target tumor, and you activate this immune system. So there are cells which are called killer cells. These are blood cells, T blood cells, which are called sometimes CD8, which means that there is a certain molecule which is called CD8, and these cells kill abnormal cells, kill bacteria. So, and with immuno-oncology, what you do, you either redirect these cells so that now they start killing cancer cells, or you inject certain antibodies which have two different affinities, one towards this killer cell and the other one towards tumor. They are called T-cell engagers. And then they bring the killer T-cell in contact with tumor cells, and now T-cell kills tumor cell. And actually, in blood tumors, it became very effective, and diseases that previously were incurable and lethal, now all of a sudden there are many patients that actually are cured. For example, in this multiple myeloma, that's the case. It's very powerful. But in solid tumors, it was not so good. We need to do something else. So I guess what else is there? Maybe these are the main directions. But of course, I didn't mention dozens of different other directions that people always try. People try all crazy things. And sometimes they work, sometimes they don't work. It's not worth mentioning it here because we don't know yet if they work. And that's the essence of science. We shoot in the dark. And most of the time we miss. Projects that I, for example, did, 95% of these projects failed. But 5%, these three antibodies, were very successful. That's what you do. You just don't give up. Unfortunately, many people, very talented people, were less lucky. And they worked all their life trying to develop drugs, and they didn't succeed. Not because they're not smart enough, just because it just happened that that direction was the wrong one. But how do you know beforehand? You don't. You just do it and hope for the best.
So in 2017, you co-founded Emuvia. What have you been working on there?
We, at Emuvia, the same thing happened, actually. We tried different approaches that I knew about before, and I thought that they would work, and they didn't. But one approach worked eventually. So we tried several. So basically what I say, I'm too stupid to know beforehand what will work and what will not work. What I can do is try a number of things systematically, and the nature is smarter, and nature will decide which of them works. And that's what we did. And one of these approaches worked very well. So that means that approach was, again, to take an antibody and that would recognize a cancer cell. So it will bind to a cancer cell. But then there's another antibody attached to it, so it's just one molecule. And that second antibody recognizes machinery on the cell that turns on cell death. So all our cells are destined to die in a very clean way, not die like anti-cancer drugs, cytotoxic drugs, toxic drugs kill, in a very crude way when cells open, spill out all the insides into the environment. It's very dangerous because some of the things actually are toxic for the body. And the way cells die naturally is called apoptosis, or programmed cell death. And that's what all cells in our body undergo. So basically, cells live in our body for a certain period of time, and then all of a sudden is a process which is called blebbing, which means from the surface of the cell, small vesicles detach, and gradually the cell becomes smaller and smaller and smaller. Then in the nucleus, DNA is chopped into pieces, proteins are chopped into pieces, everything is packed into these little vesicles, but everything is inside, so it's not spilled over. It's not dangerous for the body. And then there are special cells, macrophages, which recognize these small vesicles and phagocytize them, which means that actually they eat them up and destroy, and that's it. And we never see that, because actually in our body, old cell is immediately replaced by a new cell, and you never know that apoptosis took place. So that's the mechanism. Also, apoptosis is a very powerful way of getting rid of cancer. It has been designed into our cells that when they become cancerous, the cells turn on apoptotic machinery. And that's why we don't get cancer all the time, because we get cancerous cells all the time, but the vast majority of them, 99.9%, I mean, it's just a number, but vast majority of them immediately die of apoptosis. Unfortunately, because of the selection and certain mutations, very rarely there is one cancer cell that escapes it and doesn't have the apoptotic machinery turned on. This cell escapes, starts growing uncontrollably, and that's cancer. There are some other mechanisms to destroy it, but that's one thing. So what we did is that we used this machinery, and we found that actually we can, and again I oversimplify, we found that we can selectively destroy cancer cells. So we inject this, so we take mice and implant human tumor cell lines into these mice. You cannot do it with healthy mice because they have an immune system and they immediately destroy it. Same would happen with me. I would not worry too much if they implant somebody else's tumor into my body, because it would immediately be rejected and killed. What we use mice which are immunodeficient. They don't have an immune system, and they accept this. So that's a good model. So, and then in these mice, we inject tumor cell lines, human tumor cell lines, and they start to grow, and the tumor grows up and up and up and up. And that's the control group of animals, where you see how the tumors grow, and you say, okay, now we have this tumor that can grow in mice. Then you take another group of mice with the same tumor, and then you inject your drug. And you see that if it's a good drug, it kills the tumor, and the drug starts shrinking, and then the tumor starts shrinking, and then maybe sometimes even disappears. or just remains very small. Maybe it's not even a tumor. It remains small maybe because it's just scar tissue. And such a drug is a successful drug. So we discovered a drug that actually, surprisingly for me actually, I didn't expect that it would be so effective, but it was rather effective and could really either completely kill or completely stop progression of several types of tumor models of a lung cancer, of pancreatic cancer, and we didn't do it in vivo because a model was not available, but in vitro on a cell line, ovarian cancer. So that's actually very interesting. I'm very excited. Hopefully we will be able to advance it into clinical trials and see if it works there. And there's a good chance.
What do you think are the timelines that could be expected in changing patient outcomes?
As I said, actually, it happens faster and faster. It is really remarkable how quickly the modern medicine advances, and simply because many talented people work there. And you don't even need necessarily revolutionary changes to improve patients' outcomes. For example, there is this disease, small cell lung carcinoma. It used to be completely toxic. Actually, 95% of patients which were diagnosed with this disease would die within several years. And 5% were typically misdiagnosed. So, and actually I know a person, that's what I knew 20 years ago, 30 years ago when I was at Dana-Farber. I knew a person who actually got small cell lung carcinoma recently, like maybe it was, what, 8 years ago, 10 years ago. And I was really upset. I knew this person would die. Well, actually, this person was treated at the Dana-Farber. They didn't do anything new, but they made small changes. For example, they irradiated his brain, so to prevent metastasis there. They did a little bit more drugs. The drug cocktail was slightly changed, and this person was cured. And 7 or 10 years later, I don't remember exactly, he is completely cured. So the benchmark typically is for the cancer. Any cancer, actually, if in 5 years you don't have this cancer, you are considered cured. And that's what happened to him. Remarkable. But now there are some revolutionary changes. And one of the first changes was done in the 70s, and I actually was lucky to work with this person, a pioneer in chemotherapy of child leukemia. So child leukemia, typically, these are blood cells that start growing uncontrollably, leukemic cells, and then children would die within several weeks. They would be just like giant test tubes in which these cells grow and grow and grow, replace everything else, and children died. So, and there were some drugs that kind of worked and could delay leukemia, but wouldn't really be curative, and then children would die anyway. Most children, I don't remember, 90% children died. So the idea was to mix several drugs together with different mechanisms of action. So one drug kills 95% of cells, another drug also kills 95% of cells, but actually 95% of the remaining 5%. The third drug also kills 95% of cells, or whatever, so to speak, but of the remaining cells. And altogether, you just wipe out all cells. And now, 80-85% of children, again, I might be slightly wrong, but approximately, of children are cured for life. That is an amazing result. So the name of this person was Emil Frey III. He was clinical director of Dana-Farber.
What do you think about some of the alternative therapies moving forward, the ones that are based on modifying cells and reinfusing them into the patient.
Yes, that's probably you're talking about immuno-oncology like CAR T cells.
Mm-hmm.
So you take these killer cells and then you start directing them towards cancer cells. So now they recognize cancer cells and then they kill them. Or for example, these T-cell engagers. So you have antibodies that have affinity towards these killer cells, just regular killer cells, into tumor cells. So they bring them together and kill. So at this point, actually, these approaches became very effective towards some blood cancers. But unfortunately, so far, they're mostly very ineffective towards solid tumors. Why? Well, I wish we knew, but actually we do know to some degree. And that, what people say actually is that the microenvironment inside the tumor is immunosuppressive. Well, I don't want to go at length at what it means, and I'm not sure that I know all the details. It's not exactly my field, but what essentially it is actually to succeed in killing tumor cells, these killer cells should infiltrate the tumor, proliferate there, so there are many cells of this type, and then kill tumor cells. So, in solid tumors, there is this immunosuppressive environment, which means that these cells are, so to speak, suffocated and die. And so killer cells don't do well in the tumor, and they don't kill it. But in the blood, because it's liquid tumor, it's just the blood, and there are many cells that proliferate in the blood. These cells are much happier because they also are blood cells, essentially, and they kill them. That's why it is effective in blood tumors, but not in solid tumors. But it may change. People work on that constantly, and they start harnessing other immune cells to kill tumor cells, and who knows? We shall see. But unfortunately, when you tweak the immune system, sometimes it overreacts, and very often. And these approaches overreact, and one of the terrible side effects is cytokine storm, which means that all of a sudden a lot of certain hormones is released, and this might be very damaging or even lethal. So that's a very dangerous thing, and it's very hard to really balance your immune system so that it overreacts. So it's either not very active, or it overreacts and you have this cytokine storm. And that's what happens in most patients that undergo this kind of therapy. But cancer is a terrible disease, so actually if you can manage it, maybe it's okay in cancer.
So could you contrast what Emuvia is working on with the current state of immuno-oncology?
Well, Emuvia, what Emuvia works on is essentially not immuno-oncology. We don't engage immune system. I don't expect any cytokine storm, because the previous generations of such drugs were in the clinic. They were totally safe. Essentially, up to a very high dose, there were no side effects altogether. And only at a very high dose something would happen, but we wouldn't reach this high dose because A, we don't need it, and B, it wouldn't be practical, it would be just too expensive. Nobody does that. These drugs of a previous generation were simply not very effective, and people started to understand why. They explained why these drugs of previous generation were not very effective. They just didn't know how to make them effective. But then eventually there were several researchers that started speculating how it could be done. Very interesting papers. So I actually took. So I understood how it would be possibly done. Developed it a little bit further. We developed our original interesting approach, and actually, amazingly enough, it worked. But again, it didn't work in the beginning. We tried many different designs of these drugs. Did an experiment, and most of them didn't work. And some of these experiments were done by contract research organizations in China. And I was very nervous. And the problem with China is that it's a 12-hour time difference. So typically, result would come to me, to my email, at 3 a.m. in the morning. So I didn't sleep well that time because I always expected results. So I would check my computer at 3 a.m. and then I would open my computer. It doesn't work again. And it was very upsetting. But then all of a sudden, with one of them, it worked. It worked not very well, but then we understood why, because it wasn't made very well. We made it better, in a better fashion, and it worked fantastic. So which one of them would work and which one of them wouldn't work? How did I know? I didn't know in advance. It just happens. Nature is smarter than me. We tried them all systematically, and one of the design worked. That's how science is done. You cannot dictate to nature. Nature dictates to you.
So do you believe that the field as a whole is progressing and that we can expect longer, healthier lives for people afflicted by this deadly disease?
Of course. You already see that, actually. First of all, expectations changed. So previously, people would say, let's cure cancer. In many cases, it's unrealistic. In some cases, it's realistic. But if you can make a cancer a chronic disease with almost no symptom, it's good enough. It's just like HIV-positive people, actually. They no longer have AIDS because they take these drugs that suppress HIV for the rest of their life, but they're not sick. They're healthy people.
Well, that's an inspiring thought. Hopefully, cancer will turn into a chronic disease and maybe even cured.
Some cancers have been cured. And also, what I completely forgot to mention, hopefully, I really hope that all our studies will become absolutely unnecessary and obsolete because of the diagnostics. Because the most problematic thing is that many cancers are diagnosed at a later stage when nothing can be done really. If we could diagnose cancers at a very early stage, you don't need many drugs. You just pick up this tumor or just irradiate it, quickly kill it, and that's it. It's not a problem to remove a tumor surgically. The problem is metastasis. When it's already spread, nothing can be done. But if you detect it very early on, you don't really need many drugs. We have tools to kill these cancers.
So the war is being fought from many sides, at the outset and then in treatment.
Right.
Well, thank you for your time and thank you for joining us, Victor.
Well, it was a pleasure.
Thank you for joining us today. We hope you join us next time as we examine and elucidate the frontiers of science and technology with the thinkers creating our future.